Usually when we talk about a medical diagnosis, there is this underlying expectation of precision. It feels almost like engineering, right?
Right, yeah, like it's a structural problem.
Exactly. I mean, you break your arm, the x-ray shows that jagged white line, and the doctor just points at the screen and says, oh, there it is, that's the problem.
Yeah, because in that scenario, the problem is binary.
Yeah.
The bone is broken, or it isn't. It is structural, it's static, and most importantly, it is contained.
But then you step into the world of oncology, and specifically the study of metastasis, how cancer actually spreads.
Oh, and suddenly that static X-ray machine feels completely inadequate.
It really does, because we are looking at a microscopic landscape that isn't just broken. It is terrifyingly strategic.
Absolutely.
And today we are taking a deep dive into one of the most lethal strategies in the human body. We're looking at why ovarian cancer is so incredibly efficient at spreading, and more specifically, why it makes an absolute beeline for the omentum.
Right, the omentum, which, for those who might not know, is this fatty pad sitting right there in the abdomen.
Yeah, exactly. It's a devastating biological process. But what makes it so fascinating from a scientific perspective is its foresight.
Yes, the foresight is incredible.
Yeah.
The cancer doesn't just break off and, float around hoping to land somewhere hospitable.
Yeah, right.
It actively, chemically prepares its new home in the omentum before the main tumor cells even arrive.
Okay, let's unpack how that is even physically possible, because our source material for this deep dive is a phenomenal 2026 paper from the journal NPJ Precision Oncology.
It is a really incredible piece of research.
It is. And our mission today is to leave absolutely no stone unturned. I mean, we want to understand every mechanism, every molecular trick this cancer uses to set up shop.
And to really grasp what this paper is telling us, we have to start by completely changing how we visualize a tumor in the first place.
Right. If you think of a tumor just as like a growing lump of rogue cells, you are missing the true threat. So I want you, the listener, to imagine that these cancer cells are essentially enemy generals.
I love that analogy.
Thanks. So before they send their troops to invade a new territory, so in this case, your omentum, they send out these encrypted care packages.
Yes, exactly.
They are like malicious little text messages sent ahead of time to sabotage the local defenses. In biology, we call these packages extracellular vesicles, or EVs.
Right. But the big question here is, how does a tumor manufacture these EVs on such a massive scale?
And more importantly, right, if we understand the manufacturing process, can we intercept them?
Exactly. And to answer that, we really need to anchor ourselves in the clinical reality first. The researchers didn't just start in a petri dish. They started with human beings.
Which is so important.
It really is. They looked at a cohort of 43 ovarian cancer patients. And to make sense of the data, they categorized these patients using their FIGO stages.
And just to clarify for everyone, FIGO is basically the international standard for staging ovarian cancer, right?
Yes, exactly.
It grades the cancer from early localized stages all the way to advanced widespread metastasis.
Precisely. It gives researchers a standardized way to say, this is exactly how aggressive the disease is in this specific patient.
Okay, that makes sense.
And when they lined up the tissue samples according to those FIGO stages, they found this stark, undeniable pattern. In the patients with advanced omental metastasis, there was a massive upregulation of a specific protein called CBX2.
So the worse the spread, the higher the CBX2 levels.
Exactly.
That correlation is interesting, but what really blew my mind reading this was how they proved it wasn't just a coincidence.
Oh, the paired samples?
Yes, they didn't just look at different people across different stages. They actually looked at 16 paired samples, meaning they took tissue from the primary ovarian tumor and tissue from the omental metastasis from the exact same patient.
And that is a crucial, elegant piece of clinical evidence. I mean, in typical cancer trials, if you compare patient A's primary tumor to patient B's metastasis, you are dealing with an incredible amount of genetic noise.
Right, because humans are just vastly genetically diverse.
Exactly. It is incredibly hard to tell if a protein spike is actually the cancer spreading or just a quirk of patient A's individual biology.
That makes total sense.
But by looking at the exact same patient's primary and metastatic tumors, they isolated the cancer's exact playbook. And the playbook was clear. The cancer is actively cranking up the CDX2 protein, specifically in the metastatic lesions.
Wow. Okay, so they see this happening in humans, but to prove causality, they had to move to animal models, right?
Right.
And the way they did this was wild. They took human ovarian cancer cells and they put them through 4 rounds of in vivo selection inside mice.
4 rounds.
Basically, they let the cancer spread, harvested the most aggressive cells, put them in a new mouse, and just repeated the process until they had this ultra-aggressive, highly metastatic subline called ES2HM.
It is a way of forcing evolution in the lab to study the absolute worst-case scenario.
Right. And to track these ultra-aggressive cells, they labeled them with luciferase.
The same enzyme that makes fireflies glow.
I just love that detail.
It's amazing, right? Which means they could literally watch the cancer spread through the living mouse over a 28-day period using bioluminescence imaging.
Just glowing in the dark.
Exactly. And here is where the role of CBX2 becomes undeniable. They used CRISPR gene editing technology to knock out or delete the CDX2 protein in a batch of these aggressive cells.
And when they put those CBX2 deficient cells into the mice, the invasion basically just stalled.
It completely hit a wall.
Yeah, by day 28, the glowing signal on those mice was significantly lower. The cancer struggled to reach the omentum. It couldn't proliferate the same way. So CBX2 is clearly the driver.
Absolutely.
But the vehicle transporting the cancer's orders is that EV, the extracellular vesicle.
Right. And understanding how a cell naturally makes an EV is, well, it is essential to understanding how the cancer hijacks the process.
Okay, so I always try to picture EV biogenesis like a microscopic biological Russian nesting doll.
Oh, that's a great way to think about it.
Right. Tell me if this tracks. So you have the main cell. Inside that cell, the outer membrane pinches inward to form a tiny bubble called an early endosome.
Yes, that's the first step.
That endosome eventually matures into a slightly larger structure called a multivesicular body, or MVB. So that is your second doll.
Correct.
Then, inside that MVB, even smaller bubbles begin to form. We call those intraluminal vesicles, or ILVs, the tiniest doll.
Exactly.
Finally, that whole MVB travels to the outer wall of the cell, fuses with it, and spits all those tiny ILV bubbles out into the body's bloodstream or surrounding fluid.
And once they are outside, they're officially called EVs. That is a perfect visualization.
Okay.
And normally, this is a highly regulated biological postal service. Cells use it to trade resources or, basic signals.
Normal cellular communication.
Right. But when the cancer takes over, it turns this postal service into a weapon. The researchers cracked open these tumor-derived EVs and found they are not empty. They are densely packed with specific pro-metastatic cargo.
Specifically microRNAs, right?
Yes.
Things like miR21.5p, miR141.3p, and miR200b3p.
Exactly. And when those specific microRNAs reach the omentum, they enter the healthy cells there and fundamentally reprogram them.
Wow.
They suppress the local immune system, alter the connective tissue, and essentially lay down a biological welcome mat so that when the actual cancer cells arrive, the environment is already primed to help them grow.
Okay, let's just pause here and do a quick 5-minute recap because this really makes you rethink how diseases operate.
Good, yeah.
So if we are tracking this, bullet point number 1, ovarian cancer spreads lethally to the omentum.
Yes.
Bullet point number 2, it uses extracellular vesicles, or EVs, as transport ships to prepare the metastatic niche ahead of time.
Right.
And bullet point number 3, high levels of a protein called CBX2 correlate with worse clinical outcomes and higher metastasis.
Exactly. That is the core foundation.
So for you listening, I want you to think about the communication networks in your own body. If a tumor can basically hack the body's postal service, what does that mean for how we traditionally try to fight it?
It really complicates things.
It does. But it also brings up a huge logistical question. Because if EVs are just naturally occurring biological mail and every cell makes them at a normal baseline, how does the cancer cell suddenly upgrade its own cellular infrastructure to manufacture millions more of them?
Right.
And how does it know to stuff them with the exact right malicious cargo? I mean, it can't just be spontaneously generating new factories.
It isn't. It is aggressively hijacking a secondary manufacturing floor. And that brings us to the actual architecture of the cell membrane.
Okay, right. Because if you are going to launch a massive fleet of cargo ships, you need specialized loading docks.
Precisely. Now, standard cell membranes are made primarily of phospholipids. If you were to look at it closely, you could imagine it as a fluid, choppy ocean.
A choppy ocean, okay.
Yeah, the molecules are constantly moving, bobbing around, highly flexible. But for a cell to efficiently pinch off a piece of itself to construct a vesicle, that choppy fluid isn't ideal.
No, you'd want something stable.
Exactly. It needs a stable platform. It needs a rigid structure.
So floating in this choppy phospholipid ocean are these solid, rigid wooden rafts. And in biology, we call them lipid rafts.
Yes.
And they aren't made of the same fluid stuff, right? They are extremely dense, packed tightly with cholesterol and sphingolipids.
And those rigid lipid rafts act as the specialized loading docks. Because they are stiff, they can physically bend the membrane inward to create vesicles much faster in a process known as ESCRT-independent biogenesis.
Let's clarify that real quick, because normally cells use a very complex multi-protein machinery called the ESCRT pathway to slowly and carefully make vesicles.
Right, the ESCRT pathway is the standard method.
But the cancer essentially says, we don't have time for that.
Exactly. The ESCRT-independent pathway relies almost entirely on the physical geometry of these rigid lipid rafts to just force the membrane to buckle and form a vesicle.
It's like brute forcing the manufacturing process.
It is a faster alternate pathway. And in these ovarian cancer cells, it is dialed up to 11.
And the researchers proved this with some incredibly elegant microscopy, didn't they?
They did.
They used a chemical stain called Philippin III. And what is so cool about Philippin III is that it specifically binds to free cholesterol and lights it up under a microscope.
Right.
Then they used another fluorescent probe called CT-beta, which specifically binds to the lipid rafts themselves.
And when they looked at the cells with high levels of CBX2, the membranes were absolutely glowing.
Wow.
Yeah, they were covered head to toe in these thick, cholesterol-rich rafts.
But seeing the rafts is one thing, right? Proving the cancer completely depends on them is another.
That is where the knockout experiment comes in. The researchers treated the cancer cells with a chemical compound called M-beta-CD.
Okay, so they used M-beta-CD to essentially act as a microscopic vacuum cleaner. What happens to the cancer's whole operation when you strip away that structural foundation?
It completely collapses.
It's just like that.
Just like that. M-beta-CD specifically strips cholesterol right out of the cell membrane. And without cholesterol, those rigid wooden rafts lose their integrity and dissolve back into the choppy ocean.
Oh, wow.
Yeah. When the researchers did this, the cancer's ability to release EVs plummeted to almost nothing.
So even if the cell had massive amounts of that general CBX2 yelling at the cell to make EVs, it didn't matter.
It didn't matter at all. Without the cholesterol rafts, the whole assembly line ground to a halt. The cancer's ability to migrate and invade in the lab was essentially abolished.
It really forces a perspective shift, doesn't it? I mean, we hear the word cholesterol and we immediately think about heart disease or blocked arteries or our diet.
Right, the usual suspects.
But in this context, cholesterol is literally the architectural lumber that cancer is using to build its invasion fleet.
Is the raw material.
But wait, if the EVs are the cargo ships and the lipid rafts made of cholesterol are the loading docks, what is actually lashing the wood together to build the dock?
That would be the scaffolding proteins, specifically a family called flotillins. The paper focuses heavily on FLOT1.
Okay, FLOT1.
Yes. If the cholesterol molecules are the wooden logs of the raft, FLOT1 acts as the thick ropes tying those logs tightly together. It maintains the structural integrity of these specific microdomains on the membrane.
And let me guess, the data shows that our enemy general, CBX2, directly controls the FLOT1 shipyard.
It does. It is a direct, measurable chain of command. When researchers silenced CBX2, FLOT1 levels plummeted, the lipid rafts fell apart, and the malicious EV text messages never got sent.
All right, let's do another quick recap here at the roughly 10-minute mark.
Sounds good.
Bullet point number 1. Cancer cells rely on ESCRT-independent pathways to make EVs faster, specifically using lipid rafts.
Nice.
Number 2, lipid rafts are these rigid membrane areas packed tightly with cholesterol. And number 3, destroying these rafts like they did using M-beta-CD stops the cancer from launching its EV cargo ships.
Exactly.
So listener, if cholesterol is the literal building block for these cancer-spreading rafts, does this change how you view cellular metabolism in the context of disease?
It definitely should.
Okay, so we have mapped the supply chain. CBX2 orders the production of FLOT1. FLOT1 builds the cholesterol lipid rafts. The lipid rafts churn out the pro-metastatic EVs.
Right.
But this brings us to what I honestly think is the most mind-bending part of this entire study. We know that CBX2 turns on the FLOT1 shipyard, but how does it physically do it? Because CBX2 isn't actually supposed to be turning things on, is it?
No, it isn't. And this is where the biology gets beautifully strange.
Yeah.
What's fascinating here is that normally CBX2 is a key member of a protein family called PRC1.
Polycomb repressive complex 1.
Exactly. PRC1 is an epigenetic regulator. Its day job, especially during early human development, is to silence genes.
Silence them.
Right. It physically compacts the chromatin, the material that makes up our DNA, so that certain genes get hidden away, just can't be read by the cell.
So it's basically a strict librarian whose job is to put restricted books into a locked vault.
That's a great analogy.
But here, in ovarian cancer, it goes completely rogue. It is not silencing, it is actively driving the massive expression of the FLOT1 gene. How does a silencing protein suddenly become an activator?
To understand that, we have to look at its physical shape. Inside the nucleus of the cell, we typically think of proteins as having very rigid, structured 3D shapes.
Right, the classic lock and key mechanism.
Exactly. But when researchers ran the amino acid sequence of CDX2 through predictive software, programs designed to map out exactly how a protein will fold into a solid shape, the computers essentially threw their hands up.
Wait, really? The predictive databases just couldn't map it?
They couldn't. They found that a huge portion of the CBX2 protein, specifically its tail, has what is called an intrinsically disordered region, or an IDR.
Meaning it lacks a stable 3D structure. Wait, so this tail is essentially just wet spaghetti?
Pretty much.
How does unstructured, flexible chaos actually accomplish anything specific in the nucleus? We are so used to thinking of biology as these perfectly meshing, solid gears.
Because that lack of structure is exactly what gives it its power. Because the tail is chaotic and flexible, it allows multiple CBX2 proteins to loosely tangle and bind together. Picture a lava lamp.
Oh, this is perfect. I can picture that.
Right. So in a lava lamp, you have a liquid environment, and inside it, you have these distinct, cohesive blobs of another liquid that float around, merge together, and then separate.
All without ever fully dissolving into the background fluid, like oil and water.
Exactly. That physical phenomenon is called liquid phase separation, or LLPS.
Yeah.
And the wet spaghetti tail of CDX2 allows these proteins to do exactly that inside the fluid of the nucleus.
I picture the IDR tail acting like a velvet rope at a VIP lounge. It grabs other CBX2 proteins, and together they phase separate to form this exclusive, membraneless liquid droplet right over the DNA, a microscopic biological lava lamp blob.
That is exactly what is happening.
But how did the researchers prove it was actually a liquid droplet and not just a solid clump of junk protein?
They used a brilliantly elegant technique called FRAP. That stands for fluorescence recovery after photobleaching.
Oh, I actually love this experiment. It is so cool. Let me see if I can visualize it for everyone.
Go for it.
So they tag these CBX2 droplets with a fluorescent marker so they glow bright green under a microscope.
Right.
Then they take a tiny, highly focused laser and literally shoot a microscopic hole directly into the center of the droplet.
Yes.
The laser bleaches that tiny spot, turning it totally dark. Now, if the droplet was a solid structure like a rock, that dark hole would just sit there forever.
Precisely. A solid structure can't spontaneously heal a bleached hole.
Right.
But because CBX2 undergoes liquid phase separation, the unbleached, glowing green molecules from the edges of the droplet simply flowed back into the dark spot like water filling a depression. Yeah, the color recovered in a matter of seconds. It proved definitively that these proteins are forming highly dynamic, flowing liquid droplets inside the cell nucleus.
Okay, so the cancer is making microscopic lava lamps in the nucleus. But why? What is the strategic advantage of being a liquid puddle instead of a solid gear?
The liquid droplet acts as a superconcentrator. By creating this exclusive VIP liquid lounge directly over the promoter region of the FLOT1 gene, the CBX2 droplet acts like a magnet.
A magnet for what?
It pulls in massive amounts of the cell's transcriptional machinery, the enzymes needed to read the gene, and traps them there.
Oh, I see.
Because it's a liquid, the machinery can move around and work incredibly fast, forcing the cell to massively and continuously upregulate the production of the FLOT1 protein.
And we know exactly what FLOT1 does. It is the shipyard that builds the lipid rafts.
Exactly.
So let's step back and look at the grand algorithm of this entire 2026 paper, because the cascade is just breathtaking. Let's do our final recap.
Okay, let's lay it out.
Number 1, CBX2 uses a chaotic tail, the IDR, to act like a biological lava lamp, phase separating into liquid droplets.
Right.
Number 2, this phase separation directly turns on the FLOT1 gene.
Yes.
And number 3, if you mutate the IDR so it can't phase separate, the whole metastatic assembly line shuts down.
That is the whole sequence. They went in and genetically deleted just that chaotic IDR tail.
Yeah.
Without the wet spaghetti, it couldn't tangle. It couldn't form the liquid droplet.
And everything stops.
Everything. No droplets meant the FLOT1 gene stayed quiet. No FLOT1 meant the cholesterol rafts dissolved. And no rafts meant the EV cargo shifts were permanently grounded.
So listener, imagine looking inside a cell and seeing these temporary liquid droplets forming and dissolving to control our DNA. How does this phase separation change your mental map of how biology actually works?
It is a huge paradigm shift.
It totally changes it. We spend so much time visualizing the cell as a solid mechanical factory, but at the highest levels of disease, it's actually fluid dynamics.
It really is.
The cancer is leveraging the physics of liquid separating and the thermodynamics of lipid membranes bending to wage war.
And if we connect this to the bigger picture, it fundamentally alters our entire approach to pharmacology and treatment.
Right, because think about traditional chemotherapy or targeted cancer drugs.
Right.
For decades, they have almost exclusively been designed like a physical key fitting into a solid protein lock.
Yes, the standard model.
You find the active site on a rigid protein, you jam a chemical key into it, and you stop the machine.
But if the engine driving this lethal ovarian metastasis isn't a solid lock, if the mastermind is a shape-shifting fluid liquid droplet, how do you drug a puddle?
Wow. How do you drug a puddle? It really points toward this entirely new frontier of medicine.
It absolutely does.
Could the future of cancer treatment be a droplet disruptor? A drug that doesn't care about targeting a specific solid shape, but instead simply changes the physical state of the microenvironment inside the nucleus.
Like adding a drop of dish soap to oil and water to break the surface tension.
Yeah.
That is exactly the kind of innovation this research demands. If we can develop compounds that disrupt the IDR, preventing that condensate from ever forming, we strip the generals of their ability to send those encrypted care packages.
We isolate the primary tumor before it can ever communicate with the omentum.
Exactly. We cut off the supply lines.
It's incredible. We started this deep dive talking about the comforting binary precision of a broken bone on an X-ray. But what we have uncovered is that the true precision of disease happens on a scale and in a physical state that we are only just beginning to visualize.
It is both terrifying and amazing.
The cancer isn't just mindlessly dividing. It is a master architect orchestrating liquid phase separation and cholesterol scaffolding to conquer the body.
But the beauty of this research is that we can finally see the architecture. And in medicine, once you can clearly see the supply lines, you can figure out how to break them.
Absolutely. Thank you so much for joining us on this undeep dive. Keep questioning what you think you know about your own biology, and never underestimate the incredible, terrifying architecture of the cell. We will catch you next time.